Welcome to Study Loop.
So part A in this question is introducing how we're activating an action potential in neurons, so that way the brain can perceive that action as a change in temperature. Part B is going to be going on about how an agonist triggers the action potential. And part C is going to go on how a single agonist activates multiple proteins to give a mix of signals. So for part A, we start with the neuron. It has a body, a long axon, and a terminal end. The proteins that we're going to look at are going to be in the body of the neuron, and it's going to be placed inside the phospholipid bilayer. So according to this, when a temperature change of 43 degrees activates on TRPV1, what it's going to do is it's going to cause a conformational change where it's going to be no longer blocked and allow access for ions to get through. If you remember, we have the sodium-potassium pump, which pumps sodium outside of the cell. So when you're opening proteins, allowing ions to freely diffuse, sodium's going to be one of the main ions that comes back into the cell. Since sodium is positively charged, the inside of the cell becomes slightly more positively charged. When you get enough of those positively charged ions on the inside, you're going to depolarize the neuron and fire an action potential down to the axon terminal. That depolarization is going to cause calcium exocytosis and neurotransmitter release into the space between neurons. So when you have your neurotransmitters released, those are going to act on a different neuron, and eventually you keep chaining neurons until you get to the brain. Since neurons are very specific based on, say, temperature or touch or some other sort of specific stimulus, this is going to be perceived as a temperature change and won't be confused with, say, a touch receptor or a stretch receptor. It's very specific. For part B, we have an agonist instead of just a temperature change. So we have TRPV3 in the cell membrane. Again, it's blocked just like the first protein, but this time instead of temperature, you have, say, capsaicin is going to bind to that protein, cause the conformational change, and allow the ions to freely diffuse into the cell. This way, because capsaicin is binding to it, it's not completely temperature dependent. It's activating on its own, say, even at a room temperature of approximately 25 degrees C. Of course, if it's hot, say at 43 degrees C, you're not going to have menthol activating all of these enzymes, but when you bump up the temperature really high, you're going to activate more of these enzymes and get a greater response being processed as heat. For part C, we're acting on multiple proteins. So we have TRPV3 will be our first one. And you also have a cold-induced receptor, TRPM8. Let me label these accordingly. TRPM8. Here we have TRPV3. The concentration at which 50% of TRPV is activated is going to be less than, we'll call this 3. It's going to be less than how much, or it's going to be more than, excuse me. It's basically going to take more menthol to activate it compared to TRPM8. So when you have only a little bit of menthol, it's going to activate TRPM more readily, and you're going to get a more cool sensation. However, if you start to really increase the concentration, you're going to start activating TRPV3 as well, and you're going to kind of get a mix of hot and cool. You're still opening the cool protein, TRPM8, but you're also starting to trigger a heat response with TRPV3. So in this way, you kind of get a mixed combination. It's kind of that cooling yet refreshing feel when you like eat mint or peppermint, something on that order. Either way, you're going to get a mixed feeling because you're triggering an action potential for both.